Okay, thank you very much for Chairman. I would like to thank the organizers to provide me this opportunity to present this talk. The content of this talk has been published in this preprint, and I would like to share slide here with you. Now I have sent you my slide by chat. Now you can download my presentation from chat window. So if you are interested in, please download and check this preprint later. Oops, sorry. And the introduction, I came from the field of biometrics, and my research field is analyzing massive genes using cutting-edge computedness method. And usually I'm using tensor decomposition as my title has said. The reason I'm using tensor decomposition is because the problem I'm doing is very old. So this means here's a number of genes, typically 10 to 4. And if we consider the DNA maturation and DNA accessibility, that this is 10 to 7. And if we consider microRNA, it is 1,000. This is the number of variables is the meaning of the machine learning or statistical study. But the number of samples is very few, typically 10 to 100. So this is so-called typical large p small n problem. And large p small n problem means there are only very few number of samples, but with many features. This means variables. And this is very difficult to distinguish between them. So for example, this is some illustrative examples. It's not related to science, but for example, so there's someone forced to classify the novel into fantasy and science fiction. So typical fantasy, Snow White, the Lord of the Rings, the Knight of the Round Table, and typical science fiction, the Star Trek, X-Men, Superman. Then you will be asked, is Star Wars science fiction fantasy? And it is obvious Star Wars is in between, so it's not easy to discriminate if this is fantasy or science fiction. Because there are very few samples here, but the content of a novel is 1,000 or more than 1,000 words, so it's not easy to classify from the contents of the novel if it is fantasy or science fiction, if there are only this kind of small number of samples. And as you could see this illustrated example, the previous slide, the number of samples is very few, and the number of features is huge. It is easy to judge if which classes each object belongs. And in this talk, I would like to propose tensor decomposition to deal with this kind of problem, and it is works pretty well. And I've just published a book on these topics, usage of tensor decomposition to this large p small n problem. And this book has been published in the second last year. I'm happy if you can read it later. And then I don't think the people here is very familiar with what is tensor. Then I would like to explain what is tensor. So just one number, this is scalar. And if we have a set of scalars in line, this can be vector. And then if we have a numbers, format is a table, this is matrix. The matrix has rows and columns. And tensor is natural extension of this line. And for example, the third order tensor has 3 rows or columns like here. So now it has ijk3 suffix. Then tensor is a suitable structure to store genomic data set. For example, we measure expression of N genes for N persons for K tissues. In this case, this information, gene expression, take at N genes, the M person, the K tissues, naturally formatted this tensor because this edge represents the persons, and this edge represents genes, and this edge represents tissues. At any point inside this rectangular represent one element of gene expression. Now, tensor decomposition is some kind of mathematical trick to expand this tensor into the product of vectors. So this is the mathematical formulation of tensor decomposition. So this is illustrated in this figure. This is Z is a core tensor. This is the representative weight of each product of vector. And the vector attribute is this is a gene, and this is the person and the tissue. So using tensor decomposition, we can decompose this xijk into the product of gene vector and human person vector and tissue vector. And we need many kind of this product. So then we have to take some of this kind of many number of product. And this is the weight of each product. Then tensor is expanded as a product of the vector with the series format. Then advantage of this tensor decomposition is as follows. So now we expand the xijk into vector of I and the vector of j and the vector of k. We can easily interpret the meaning. For example, if we don't know which tissue is important, for example, which gene is expressive only in one tissue, and which tissue is it, then we can simply check this vector because this vector represents tissue dependence. And if we would like to, if the gene's expression is distinct between healthy control patient, you can check this, and this is attribute the person. And if this vector is expressed only for healthy control, this means this express some genes. It is not expressive in patients. Then checking which pair of product has more weight for this product. Then if this vector has more larger weight, this one, this is weight, and we select some vector represent the distinction between healthy control patient, and some vector represent some tissue specificity, then we can check which G has the largest value with this which vector, and we can, if we check this vector, which gene is more consistent with this person dependence and tissue dependence. Now, I would like to apply this simple mathematical trick to drug repositioning of COVID-19. So, as I have told in the beginning of the talk, this has been published in the preprint here. If you are interested in my talk, you can check this my print later. So this I have already uploaded this presentation slide in chat box, so you can download this slide and you can check this paper later. Now we prepare some kind of gene expression profile taken from this public database. The number of genes, this number, are almost 20,000, and J represents samples. In this case, sample represents the 5 cell line. All cell line taken from the human lung because SARS-CoV-2 infected the human lung. And then K represents the comparison between the infectious cells and not infectious cells. And M represents biological replicate, how many times this measurement has been repeated. Then G expression of expression of ICE gene at JS cell line, if it is infected or not, and which biological replicate is represented here. And now it can expand the cell line vector, and this is a vector represented if it is infectious cell or not. and this is a virus replicate, and this is a gene vector. Then we can check these three, and here, so this is a result. So this is a cell line vector, and since we'd like to find genes whose expression independent of the cell line, so we'd like to find which vector is independent of cell line, and this is K. K represents if we infected or non-infected. Now we would like to know which genes are distinctly expressed between infectious cells and non-infectious cells. So this should be signs reverse. And the gene expression should not be dependent on virus replicate. Again, we'd like to have constant. Then we have found the first one constant value, and the second one who attribute to the K is distinction. Then 1, 2 it should be selected, and here G is first vector. This is attribute to the cell line should first because it is constant, and the second one should be a second vector because it is distinct between infectious cells and non-infectious cells, and this third vector should be first because independent of cell line. Replicate. And we don't know which this gene vector associate with this 1-to-1 pair. Then we found this is a 4, L4, this one. Fifth one has largest weight with these three. Then we know that this vector should be important. Then now next, we'd like to find which gene is most expresses this vector. In order that we assume this vector, gene vector, obeys the Gaussian distribution. And then if this is Gaussian distribution, the p-values can be attributed as a chi-square distribution. This is the cumulative chi-square distribution, and this is a standard deviation. This is so-called z-score, and scale z-score can be used to attribute p-values to the ice gene. And this ice gene, p-values attribute ice gene collected by Benjamin Hohenberg method because the number of genes as I've told you, 20,000. So this is a heavy large p small problem, and the number of variables p is 10 to 4, but number of samples only 30. So we have only 30 samples, and using this we would like to find which drug is effective for SARS-CoV-2. And purpose audience here, such kind of tests should not be possible because we have only 13 profiles and the huge number of drugs, so it is not easy to predict which drug is effective to SARS-CoV-2, this kind of small size problem. But we can do this. So first, this sigma 5 should be optimized because if we compute standard deviation using all U5I, this is the so-called p-value, histogram p-values. And if the hypothesis completely fulfills, histogram y minus p should be flat. And if we optimize sigma 5 as much as possible to be a flat, this histogram 1 minus p looks like this. So this is a region which obeys the hypothesis. This means Gaussian distribution. And this sharp peak for 1 minus p. So this is small p, and this part can be regarded outliers, and it should be selected, which is consistent with infection of SARS-CoV-2. Then we could select this criterion if we compute p-values and correct, and if we select genes whose collected p-values are 0.1, we can select 3067 27 genes. And so interestingly, I'm not sure people here are very familiar with this kind of problem, but there are several methods, some kind of state-of-art method to select genes which is distinct between infectious cells and non-infectious cells. If we try simple t-test and correct p-values, and if we require corrected p-values less than 0.01, surprisingly, almost all genes are judged to be expressed differently expressed between infectious cell line and non-infectious cell lines. And this is some kind of method is adapted to the gene expression profiles. It is also we cannot select. I have done something wrong. So sorry. No genes are selected, and some could not select no genes here. And only Lima has select reasonable number of genes. Then D-sect2 is a more optimized method, but as you can see, the dependent cell lines, this number largely fluctuated. So, and so our method can find reasonable genes. And in order to check if our selected 3,000 genes are correct one or not, we checked if these genes are overlapped with genes which known to interact with SARS-CoV-2 protein. This is a list of SARS-CoV-2 protein, and this is a minus log 10 p-value. So this is the surprisingly small p-values. So this bar is our method, and D-sect2 this is a state-of-art method to select genes. And here, compared with D-sect2, our 3,000 genes surprisingly well select genes which are known to interact with SARS-CoV proteins. So our method works pretty well in spite of the heavily large p-small problem. Then we can select drugs using this data. How we can do this? So it is very simple. So we have selected promising 3,700 genes, and we will try to which drug target these genes. And we have there are several databases, but we have used in this case the G-Express omnibus based drug databases. And for example, this drug immunity has 442 genes whose expression expressed with the treatment this drug, and among these number of genes, 316 genes are selected, included its genes we have selected. So as you can see, so using our 3,600 genes, we can have many drugs which is supposed to target these genes. And if then we don't know it is correct answer or not, because this is very simple procedure. It is unlikely works. So it works pretty well. Sorry for this number is reference of preprint. So if you are interested in which paper has been reported these things, please check my preprint later. But as you can see, for example, this one, this is top drug predicted by a method, has already tested by COVID-19 some time ago. And I have no time to explain drug one by one, but we have found at least one difference which report these drugs once tested to drugs of COVID-19. And this is a databases. Gene expression has been downregulated the treatment. And this is another part. This is a drug whose treatment and upregulated. And upregulated genes, if overlap or not, this here. So for example, this drug has 360 downregulated genes, upregulated genes, and among these 276 genes are selected by our 3,600 genes. But again, we have found all compounds once tested SARS-CoV-2. This one is not really SARS-CoV-2, but SARS-CoV. But as you can see, and please check the references if you are doubtful these outcomes. So this means our strategy can correctly predict which drug are effective in the disease simply using only 13 GX profiles. So this means our method can effectively work for large P small problem, and our method can predict drug repositioning very well. So I have not studied that, but this means if we check whole list of, I did not present here the whole list, but if we check whole list of genes, drugs predicted by a method, possibly it should include many promising compounds which has not been tested but might be effective in SARS-CoV-2. The conclusion. We have applied tensor decomposition based unsupervised feature expression with the optimized ST. This is sorry the lengthy, but this is the name of the method we have used. This is the general method which can be applicable to any kind of GX profiles. And we have used this method SARS-CoV-2 drug repositioning. We have selected 3,729 genes, whose expression is distinct between the infectious cell line and non-infectious cell line. And we tried to, these genes turned out to be heavily overlapped with the human gene known to interact with SARS-CoV-2 protein. So we have our method correctly select the genes which should be important in the infectious process of SARS-CoV-2. Then if we check which drugs target these genes significantly, we can list promising drugs. Almost top 10 drugs predict this method once tested to SARS-CoV-2 drugs. So this means our method is very promising strategy. And the point is we don't need a large data set. We have only 13 GX profiles. Then we can predict drugs very well. Thank you very much for your interest.